Okay, Jose. So a lot of people are asking about what type of samples can be run on the Tapestri platform.
Good question. Yeah, so the Tapestry platform is compatible with most of the sample types, such as the cell lines or frozen, cryopreserved sample types, such as PBMCs or bone marrow cells, as long as they are viably cryopreserved. When it comes to solid tissues, Tapestry also enables users to dissociate nuclei or isolate nuclei from frozen tissue section or fresh tissue section using our nuclei isolation protocol, making that also compatible with the platform. The only other main question that we get from a lot of users is the compatibility with the FFPE. Right now, it's still in early investigation phase where we haven't shown any compelling data to show FFPE works as well as the other sample types.
Okay. Well, it's great that it sounds like, from cell lines, PBMCs, that the Tapestry workflow is compatible. When you talk about working from tissue, how much do you need to do the nuclei isolation protocol?
So the tissue, quantity depends on how nucleated particular sections of nuclei you're working with. A heavily nucleated tissue, you may need, for example, less than 10 millimeter cube. And something that doesn't have sufficient amount of nuclei, you may need, much more than that. Or OCTs or tissue sessions from a slide, definitely as long as you can produce or generate about 100,000 nuclei, that is what's required as sample input into the Tapestry workflow. As long as a nuclei section allows you to produce that, then you're ready to start the workflow.
Okay. So what if a customer doesn't have 100,000 cells or nuclei? What can they do? What are their options?
Yeah, again, the ideal recommended range for the platform is 100,000 cells. However, there are some flexibility where if you're slightly lower, the outcome of that will be you may on the back end will get slightly lower cell throughput. When we say the range, customers have comfortably used, as low as 50,000 cells. And then once the workflow is done and when they're analyzing the data, the cell throughput on a typical 100,000 cell input, we get about up to 10,000 cells bioinformatically identified. Whereas if you're on the lower side of things, that will reflect on your cell throughput. The other part of it is that if you're too low, let's say you're about 10,000 cells or 10,000 to 50,000, so that is not any recommended range. Definitely there's a higher risk in terms of not having enough template for the single cell biochemistry to work. So there are some flexibility in the 100,000 number, and that's all about getting how much cell throughput do you require for your particular experiment.
Okay. And so I guess if a customer is willing to take a cell throughput or output of under 10,000, so then it sounds like that they can work with less than 100,000 cells?
Yes, correct.
Okay, great. So I've seen data from the Mission Bio platform that uses sort of spike-in experiments and titration experiments. What type of cell lines are used in those types of experiments?
Yeah, good question. So these cell lines are very well genotyped with other orthogonal methods where we do know the truth or the genotypes at, where we are covering these particular, mutations of interest. So we're using these cell lines such as K562 or Raji, where there is existing data sets, so using that information when we are actually going to spike in such cell lines into our samples of interest. And when we finish the workflow and analyze the sample bioinformatically, we can identify these particular small spike in populations of cells based on the genotypes that we know of from your actual samples. So we spike in any known cell, genotype, very well genotyped cell lines, and the platform has shown compatibility to identify populations as low as 0.1% very consistently and robustly.
Okay, great. So I know many of our customers use FACS or MAX to process their samples or enrich their samples. Can those types of enriched samples be used on the platform as well?
Absolutely. So, there's I would say a significant amount or significant percent of our customers do like to study particular populations of cells or underlying genotype for the particular populations of cells. So they do use MAX to enrich for certain, cells based on what they're expressing as surface markers. And as long as the MAX or the magnetic beads are, relatively small in size, such as less than 2 micron, if your cells are bound to such small beads, you can input those cells into the Tapestry platform, and there won't be any underlying issues or, and workflow works smoothly. However, if you're working with magnetic beads that are more than or larger than 2 microns, we would require you to, elute the cells from the magnets or do a negative selection and then use cells for subsequent workflow. For FACS, again, same idea. A majority of our customers are interested in, for example, when they're studying leukemias, particularly AML, people are interested in FACS sorting for blast cells, which they run on Tapestry to understand the underlying clonal, architecture for those blast cells as well.
Okay, great. So with these heme applications, is it possible to use blood as an input for the Tapestry platform?
So we generally recommend customers, if you can use, depletion methods to remove any red blood cells from your samples. It will be best in terms of, the chemistry. However, we do not think blood may necessarily interfere with the workflow, but we strongly recommend working with depleted, especially red blood cells, depleted sample types, such as PBMCs or BMCs, using a standard FICO or other gradient separation.
Okay, great. There is also a question just around the size of the cell that can be accommodated by the Tapestry droplets.
Yes, so the Tapestry platform, has successfully worked with samples, primary cell sample types that are as small as 2 micron, 2 to 5 micron, and we have, shown cell lines that are 20 microns or up to 25 microns can be processed with the platform with no problem. So we would recommend that if your cell size is within the range of 2 micron to, let's say, 25 micron or a max of 30 micron, there should not be any issues. So that's the range you want to aim for.
Okay. Great. And for the attendees that have joined us, if you are interested in asking a question, please, again, feel free to ask it in the Q&A box or in the chat link for the Zoom. So maybe I'll move away from the sample prep questions, Jose. And I know last week we heard from product manager Jim Flynn around the Tapestry Designer. And I think he gave a great demo of it. But if I'm a new customer, how would I go about getting started on the Designer?
So all our bioinformatic tools is easily accessible on our website called Tapestry Portal. So Tapestry Portal has our one of our bioinformatic tools called the Tapestry Designer, which our users use to design their target NGS panels that they use for Tapestry. And also in the portal, you can find other bioinformatic tools for downstream analysis as well. So as a first step, going to Tapestry Portal, signing up as a user, then you can access our Tapestry Designer. And then in the Designer, you can see what are our catalog standard panels that are available. And as well as you can get a feel of, how the design process works, or, what is the content of the panels, and etc.
Okay, that's great. So with these custom panels, how long does it typically take to create, manufacture, and then ship a custom?
Good question. So the creation process is like, all you need as input is to know the locations of interest, either, your a cosmic ID, or if your gene of interest, a name of gene of interest, or, coding chromosome coordinates. If you have such informations, that are ready, you can input such information into our Tapestry Designer, and then in a short period of time, you will get a designed panel ready, and it kind of gives you an idea about what are the coverage for the panel, how well this panel is represented, and if that is what you are looking for, and if it meets your requirement, you can proceed by pushing it towards manufacturing. If not, you can work with our design team to further improve the content of the panel you designed. Once it's in the manufacturing, it takes up to 2 to 4 weeks for a regular custom panel. However, there might be special requests and a larger panel that are beyond our typical multiplex capability. If that could take more than 4 weeks, but a typical turnaround time after it's in manufacturing is 2 to 4 weeks.
Okay, 2 to 4 weeks, great. When we think about the size of our panels, what's sort of the minimum and maximum number of amplicons that we can accommodate?
Good question. The Tapestry single-cell workflow, relies on a multiplex PCR approach. So in order for that multiplex PCR to work efficiently, we have a minimum requirement of amount of amplicons. So 20 amplicons in the panel is our lower limit. And as an upper limit, you can go up to 500 amplicons in your panel. However, if you would like to go beyond 500 amplicons, you could work with our design team to see how much larger your panel can be designed to be. But 20 to 500 is our default.
Okay, great. And what kind of QC exists for the designs that come from the Tapestry Designer?
So once the targets are inputted, we provide you information such as how well particular regions are covered. We conduct an in silico test in order to identify whether these primers are, how efficiently these primers are going to be binding, in wet lab testing. So there's a prediction mechanism there. And if there are GC-rich regions or regions that we think are going to have some primary inefficiency, we provide you an output saying, hey, these are potential variants that may not perform well. So you get an idea within a typical panel how well that might perform after it gets synthesized. So yes, coverage metrics kind of telling you how well your amplicons or targets are covered, and other QC metrics as certain regions being GC rich, etc. So you get a few output files that allows you to decide whether the panel is well represented for your project.
Okay, great. And if I'm browsing on the designer, it looks like there are some catalog panels, and then there are also some pre-designed panels. Can you help us understand what the difference is between the two?
Absolutely. So catalog panels have been, used by our user base, and it's wet lab tested. It's ready to purchase. Whereas this pre-designed panel is out there as a template for users to use as a reference. And if they want, they can proceed towards submitting it as a custom panel. So these are not ready to purchase or already built. These are just a template that if you're interested, you can take regions of interest, or you can go along with as it is designed. So that's the main difference, where catalog are fully made and that are ready, whereas the pre-designed are just a template for you to work with and submit it if you're happy with the content.
Okay. What if I wanted to say take a cross of an AML panel and another design from a pre-designed panel on some of my own unique content? How would I go about doing that type of thing?
Absolutely. So every panel, has a data sheet associated to it, and we also provide an output file called a bed file, where we tell you what are the chromosome coordinates for these amplicons, what are the start and the end, and we provide you the name of the amplicon. So any catalog panel, you go through the list, and you can pick out a few ones you like, or you can pick the entire content itself, and you can actually add onto it. So you can browse through the other pre-designed panels, and if you like other amplicons in there, you can add to that list, and thereby you can create your own custom amplicon panel, and you can submit that panel that might be a cross between multiple different existing panels.
Okay, great. And so where would I get all of the contents of these catalog panels? How do I see what's in them?
Absolutely. So the way to get them is once you go to tapestryportal.com and you go to Tapestry Designer, one of the icons there is browse for catalog panels. And once you look at our catalog panels, for example, we have a AML catalog panel, and clicking on the AML catalog panel gives you opens up a page in which where you can download the associated files that are for individual panels. So you can get the content from there itself. And in terms of content, you can get a bed file which tells you the regions itself. And we also have associated data sheets where it tells you hotspot mutations that are being covered by this panel as well.
Okay. And where can we get the data sheets? Are those also in the same place?
Exactly the same place. If you click on a catalog content panel, it opens up the page, and in multiple tabs, you can see whether if you want to just download, a file associated to it or a data sheet associated to it. All those are also, deployed in the Tapestry Designer itself, and so you can easily gain access to our content.
Okay, great. I know one of the other exciting things that Mission Bio is talking about these days is the capability to do both DNA and protein from the same cell.
Yes.
Can you talk a little bit about maybe the overview of what that workflow looks like?
Absolutely.
Customers have
So
the same sample, or what's that like?
absolutely. So, our first product has been, targeted towards amplifying selected regions in your DNA using a targeted NGS panel. So, in our user base was definitely requesting if we can layer on top of genotype information a way to understand the phenotype. So we listened, and then we decided to come up with another assay, in which the instrument doesn't change. It's just the assay side of things or wet lab side of things outside of the instrument changes. Where for a typical Tapestry workflow for your DNA only, where you prepare your cells and input into the cartridge that goes in the instrument, for the DNA plus protein side of things, the sample prep stage, you stain your cells with with a set of antibody oligoconjugates of your interest. So we provide you a menu of existing antibody oligoconjugates, such as CD33, 34, 3, a list from which you can choose a few markers. You can comfortably choose up to 15 markers for your assay, and then you stain your cells with these antibody oligoconjugates before you input your sample into the Tapestry workflow. And within the workflow, basically, we capture these cells that has, these antibody oligos or the mix of antibody oligos, and they are still, captured as single cells in single droplets. And our workflow works in such a way that your DNA template gets amplified and a cell barcode is incorporated. Similarly, within that droplets, all the antibodies will also share the same exact cell barcode that are shared by your DNA target. And then afterwards, following through the standard library preparation and sequencing, you can see from within which cell, what underlying genotype information or genetic landscape is, as well as what are the expression patterns for these antibodies that are within your, or surface markers that are within that particular cell. And then when you're doing the data analysis side of things, now you have the ability to visualize multiple different clones that are either defined by just genotype, defined by just the expression pattern of proteins, or defined by both. So this gives you that extra layer of information, whereas you could select for populations, let's say, a CD3 population or a T cell population that you wanted to understand certain mutations. And with Tapestry's multi-omics approach, you now have that capability where you can look at a particular population or cluster of cells that are defined by the expression of certain antibodies, and you can understand the genetic mutations of those cells in that population.
Super exciting stuff, very cutting edge. In terms of the workflow for Tapestry, what type of additional instrumentation or items are needed in the laboratory workflow?
Yes, good question. So the Tapestry instrument is a droplet generator, as well as where our cartridges sits and where we generate these emulsions, capture all the associated content to do the library preparation. So other equipment such as I mentioned in the beginning, we do a multiplex PCR in order to generate these libraries. So you do need access to a thermocycler. And then once these libraries are, processed, in order to look at the quality for these NGS libraries, you need additional equipment such as, a bioanalyzer or tape station, or to look at the concentration, a qubit or a qPCR. And then these libraries are ready to be sent for sequencing on any of the Illumina sequencers. And then in addition to that, just the standard library prep reagents, and our user guides goes into the details about what those excess reagents are. But that's about it.
Okay. So if a customer is already running NGS libraries, they likely have everything that they would need.
Most likely, most of the customers before we go on site for a training, we reach out and we go through a checklist. And most of the customers who are NGS users has most of the equipments already in the lab. So they're just the basic additional, requirements for equipments.
Okay, perfect. Okay. Well, maybe just a final question, maybe we can tease what might come like next week for some of the more pieces around after you've run the Tapestry platform, what comes next? So when the data comes off of the sequencer, what does the customer do with it then?
Yes. So, the Tapestry platform, we provide an end-to-end solution. After sequencing is done, our users have access to a pipeline. We provide them, an account in our existing cloud-based, pipeline, where users can use the pipeline to process the FASTQ files. So once the sequencing FASTQ files comes through, and then they can upload these FASTQ files into their pipeline account, in which we do basically two things. First, we map these FASTQ files into using our genome reference, which is a human HG19 build. And after these FASTQ files are mapped, we identify our cell barcodes, calling the cells, and then we use a haplotype caller called GATK to call the variants associated to your data, to your samples. And then we provide you some output files for you to do some downstream visualization. So we provide you a file called VCF, which is your variant calling file, and a similar output file, which you can visualize into using another software that we provide for free. It's a desktop deployable software called Tapestry Insights, which you can download from our portal. So you can take this output file, which comes out from the Tapestry pipeline, and then you can visualize the clonal architecture using bar plots or fish plots or things along those lines. And we have tutorials and additional guidance and documentation also on Tapestry portal for how to use our Tapestry Insights to visualize the clones.
Okay, great. If anybody has any questions, again, here's your final chance to get them into the chat or the Q&A. Okay. If not, I do want to thank the attendees for joining us today. And Jose, thanks very much for answering our questions.
Absolutely. Bye-bye.
And join us next week for office hours with the support team. Thank you.